So let's start.
Let's start?
Yeah.
Okay. So we don't wait for Professor Arvind.
No. So
we can start. Okay. So good morning, everyone. So it's lecture number 6. We're almost halfway through this series of lectures. I'm reminding you we're discussing the role of stochasticity, or randomness in chemical biological processes, and what we do is we're learning by examples. We look at different systems where stochastic models are applied, and this way we are learning how to roll stochasticity. Okay, so again, I encourage you, feel free to ask me any questions, and we will have time after if so this will be the story on proteins target search for specific sites on DNA. All the stories are biological. Whatever left, everything is biological, so don't worry about it. There will be no chemistry. So protein-DNA interaction. So as you know, protein and DNA molecules are two main classes of biological molecules, and essentially interaction between them govern all processes in living systems. And many biological processes have to start after proteins, specific proteins known as transcription factors, bind to specific sites on DNA. So the example is transcription. So transcription is a copying DNA, creating exactly identical DNA, and it's done by it's creating RNA molecules with the help of enzyme RNA polymerase, which just reads it and creates it. So this is the blue one, is RNA polymerase, which is supposed to read DNA, create the corresponding RNA copy. The problem is that RNA polymerase doesn't know where to start, and so the transcription factors, other proteins, bind. For example, in this case, there is a sequence called TATA, nothing to do with TATA family. It's TA, 25 base pairs from where you have to start transcription. These transcription factors bind, recruit some other proteins, and then RNA polymerase can properly start and read the DNA. That's because DNA, in some sense, is a set of words, like sentence, right? But you need to know where to start reading, because you can read something wrong and you will get wrong message. So that's the point. So the interesting thing here is that this target, the sequence to which the transcription factor has to bind is relatively small, order of between 4 to 10 base pairs. So it's a order of 1 nanometer. While DNA is long, it's huge. Human DNA has 1 billion, more than 1 billion base pairs. So this is a very difficult problem. It's a problem of search. You need to find something very small in a very long object. So, so this is my analogy here. Imagine that you're driving from somewhere, from Delhi to Bangalore, and you have to find some village in Maharashtra. That's a real scale. Like for the first, you have to understand it. For the first time in your life, you have to find something which you never been. But that's how difficult the search. This protein's target search, you can also, there is a lot of interesting observations. So that some of the proteins, for example, this protein called lac repressor, they actually, people measure it, and the rate how fast this protein can find the target is amazingly fast. So in this case, it's 10 to the 10 per mole per second. Okay. The problem it's not that it's so fast, although it's a second, bless you. Also, it's not the second fastest chemical rate. The problem is that it violates some theoretical limit known as the Device-Mulhovsky diffusion control set. So remember we said when we talk chemistry, right, that reaction happens after collisions, right? So no reaction can happen at least until one collision occurs. But this reaction apparently happens faster than collision. That's a miracle how it can happen.
So this allicate protein that was already along the RNA, or
no. These proteins only have to bind something on DNA, and that will stimulate other things, transcription, whatever. But it has to bind somewhere on DNA. So the rate of binding is very fast, extremely fast, and it violates this limit 100,000 times faster. This phenomenon is known as a facilitated diffusion. It's still, to some sense, controversial. So this is my very politically incorrect, naive explanation. If you don't like someone, you want to hit someone, right? How fast you can do depends how fast you can approach this person, right? But apparently this happens before. So this is Klitschko, famous Ukrainian boxer. Anyway, so this is the point. For reaction, you need to collide, but reaction happens faster than collision. So that's what is strange here. Yeah, so well, you probably know this, but let me remind you. So this is when reactions, very fast reactions, they so-called diffusion control. Diffusion control means that the rate limiting step for reaction is how fast two molecules can approach. And there was a theory developed by 100 years ago, and theory goes back to two famous people, Debye and Smoluchowski. And according to the theory, if you have molecule A and molecule B, if you want to estimate the fastest, the highest possible rate, you can do this analysis, and this is the result. So the maximal possible rate is sum of diffusion coefficients A and B. 4 pi is a three-dimensional geometric factor, and the R is the distance, essentially sum of the sizes of the molecule. So if you can put, we know typical proteins diffusion coefficients. We do back of the envelope calculations. That should be the maximal possible rate. But that's what I told you 2 minutes ago. Experimentally, it's 100 times faster. So there is something miraculous about this facilitated diffusion. And by the way, there is a diffusion control rate, and there is additional correction comes if the molecules are interacting through long range. There is additional factor comes if the molecules interact through some potential of interaction U, and there is additional factor data which is expressed this way. So the formula, this formula is with no any long range interaction. This formula is a generalization if there is a long range interaction. Yeah, so there are two names here I mentioned, Debye and Smoluchowski. So Peter Debye, Jewish American scientist, Nobel Prize winner in chemistry. So although he did spend most of his time working in Holland, Netherlands, and Germany, the last 25 years he was in United States, and actually he was in the department from which I graduated. That's a Baker Hall in Department of Chemistry at Cornell University. And by the way, his windows was over there. What is interesting, when I was a graduate student, I was sitting in the same room. Okay, his office then was divided and given to graduate students. So I was sitting in one of the rooms where Peter Debye was sitting before. He died in 1966, so it's before I was born. So I didn't see him, but people still remember Peter Debye. He was one of the most universal scientists. Did a lot of things and many things, and we actually will see Debye again in another lecture. Anyway, the second person was also very interesting. Marian von Smoluchowski, from very rich Polish-Austrian family. Yeah, he's a Polish-Austrian scientist. People ask me about Brownian motion. He is the one who did the papers on Brownian motion along with Einstein. He investigated the random motion of molecules in liquids and gases. Was a very talented musician, painter, mountain skier, mountain climber. Unfortunately, he died of dysentery. So wash your hands before and after you eat, okay? Good. So let's come back to protein target search on DNA. One of the dominating theories in this field goes back to a series of work which come from Berg, Winter, and von Hippel. And okay, so the idea was how we can explain that this search is so fast. That's the idea, right? Why it's so fast. And those people argue that it's actually, so my comparison is not exactly correct. So let me try to remind you. My comparison was purely three-dimensional through the collision. But the people argue it's not exactly the right things. What's actually happening, the proteins occasionally bind non-specifically on DNA, scans DNA, dissociate, come back. So it's not pure 3D, it's a combination of 3D plus 1D.
So this K only is the site dependent, like
in principle, yes. For simplicity, you can neglect it. Yeah. So the people arguing here that acceleration due to lowering of dimensionality, and you probably know that if you are in dark room, but if the room is like this and you have to find something, you can find eventually, right? But if you're in three-dimensional object, you don't know where to go, you are lost. So they are arguing lowering dimensionality, which helps you to find things faster. But then if you think a little bit, this is absolutely wrong argument. This is argument of probability. So the probability of finding something may be more, but it doesn't guarantee that you reach quickly there. And rates are its inverse time. So it's not exactly the right way to do it. Okay, so this is, I'd like to know that this is so-called thermodynamic argument, or in mathematical language, it's a probability argument, while I was trying to explain dynamics, how fast you can reach something. The problem is kinetic or dynamic. Good. But this approach allows you to have a very simple formula for time to find the target on DNA. So L is the total length of your DNA. Lambda is this average size that you scan on DNA in each search cycle. And this is time to be on DNA, time to be in solution. So it's a very reasonable thing. So L over lambda is the number of cycles you have to do before on average you will find the target. And then the analysis shows that you can actually reach so-called optimal search when the time to be in DNA in solutions are the same. And in this case, you can really kind of accelerate of orders of 100 times. Very similar to what they see in experiments.
Excuse me, Professor. Is this the theory you talked about on the previous slide, the 3D excursions followed by the 1D sliding?
Yes, exactly. This is called 3D plus 1D, yes.
Did you say that this is not sound because it...
Yeah, I will show you it's wrong in a few moments, but this is very commonly described in textbook and absolutely wrong. It's absolutely wrong. People are mixing probability and dynamics. People subconsciously think if something is more probable, it will happen sooner, but it's generally not guaranteed. Okay? We all die, right? The probability is 1, but we don't know how long it will take, right? Anyway, so this is essentially these people try to put this explanation. So they think, okay, so first of all, there is So people who put this idea, not mathematicians, some of the physicists or chemists, and they don't know how to solve differential equations with the proper boundary conditions. They screwed up with the boundary conditions. But let's at least explain the physics. So the physics, the people think, so the point again, the idea is why some of these proteins can find the target so fast. And the argument is, oh, that's because all these proteins need to find the region of this lambda cube, and then you'll find target eventually. So if I go back to this Debye-Smolkovsky formula, they're saying the presence of DNA effectively increases this size. So the target is not small anymore, but a bigger one. Okay? That's what they say without doing mathematics. But of course, this is wrong argument because you can come to this region and then go away. It's not guaranteed that you come back to target. Okay, so let's try to be now, let me try to be critical and show that what they're saying is nonsense. So they typically assume diffusion coefficient in solution DNA is the same, and the time to be in solution DNA is the same. Okay, so this is the only way to have a relatively fast search, but experimental conditions show that it's very different. You are very slowly moving on DNA, and you are mostly on DNA, and if you put these experimental conditions into this theory, you will never get acceleration. You always get worse. So it's so for example, this protein P53 that I mentioned, there was experiments looking at this P53 moving on DNA, and they found that 99.9% you were on DNA. And if you're very slow on DNA, how the overall process can be fast? So it's not correct. And there's even more. This comes from physicists because chemists would never do it. So these people argue, so they created some flaws, and then, so this is prediction of rate as a function of some concentration. So if what you need to look at this blue line. So this is according to this blue line, a specific set of conditions. You change the concentration of proteins, you decrease the concentration of proteins, and reaction goes faster. Nonsense. Nonsense from the point of view of chemistry. You dilute gas in your car drives better, right? No way. We argue that chemical reactions due to collisions. This is physicists. Unfortunately, physicists don't have chemistry. You have to take chemistry sometimes. So it's a wrong result here. Good. And then comes another group of biologists who also completely do not understand something. And they say, oh, there is any problem that proteins can find target fast. That's because everything is done in electrolytes. And you remember electrolytes, charges interact, Coulomb interactions are long range, and they say this is long range things here. And so for this reason, you move things faster. If things are attracting each other, you can move faster. Problem comes, they forgot about something which is known Debye length. Again, the same Debye did something different. So Debye length is the length over which, so below this length, electrostatic forces are important, above is not. In a system where many charges compensate each other, they screen each other. So there is a size of a system which depends on the total number of charges, below which, for distances below this, electrostatic is important, but outside is not because of the cancellation. And for this system, the Debye length is very, very small. In other words, in these real biological systems, electrostatic doesn't play a role. For the search, electrostatic is important, but it doesn't play the role. Again, this biologist who probably didn't take chemistry at all, he didn't know about the electrolyte. Good. So, but there are more problems, more theoretical problems. And the reason I want to show you, because this is an example of something that we discussed a little bit yesterday, continuum versus discrete. Continuum is a special case of discrete, but if you're not careful, you can really get some nonsense. And that's an example of this nonsense. So the people use mostly continuum theories here. And this is the result of continuum theory, essentially solving differential equations, the continuum theory. So let's look at this formula. So this is the time to find the target. L is the length of DNA. Lambda is average distance that DNA non-specifically scans DNA in one cycle. This is time to be on DNA, time to be in solution. Time to be on DNA, you can write it this way as this distance, lambda square over 2d1, right? Seems reasonable, everything reasonable. So let's try to do suppose my diffusion coefficient of DNA goes to 0. And this formula predicts infinite time. It will blow out. You divide it by 0. It's infinite time. Okay? But that's unphysical. It's not possible. And because the proteins can bind and unbind. Proteins don't need to scan the whole DNA. And then there is a real system. There is a real proteins which do not slide and still find target quite well. So this is a protein called REK, very important proteins. So our DNA, we're constantly under threat on the sun and so on. That's why probably doctors say you have to use sunscreen, sunlight, whatever, a lot of oxygen, reactive species break some DNA, okay? And then there are proteins which try to find these issues. One of them is REK. So it has to find problem on DNA, some chemical incorrect insertion quite fast enough. But these proteins do not slide on DNA. So they come, unbind, diffuse, come, unbind, and they do it in finite time. But according to this theory, it would be infinite time. This is an example of application of continuum theory where you cannot apply continuum theory. Okay? Good. There is additional problem which is known as a speed selectivity paradox. Speed selectivity paradox. So DNA is a heteropolymer. So when something moves on DNA, it's not a smooth ride. Molecules interact differently with different bases of DNA. So it's a very noisy environment. So this is a typical potential. Then people argue that in this potential, the diffusion coefficient should be quite low. The formula goes back to 1/6, for example. But you cannot do it if the potential should be smooth enough for you to move fast enough. But in reality, the system is very rough, but you have to assume that it's smooth, so there is contradictions here. So this is my analogy. So if you drive in India, as you know, right, sometimes it's horrible traffic, but sometimes it's nice. So on average, your time will not be very fast if you are. So that's the point. Okay, so this is the problems with current use. There's a fast search, but you don't know how to explain it well, because if you, that's not me. Updates come always on time, right? Okay, good. So these are the problems. You cannot explain reasonably well fast search. People use continuum models. And you have to have a smooth potential in DNA, but it's probably unrealistic. And there are some sort of things. So I would like to show you that stochastic picture can explain the things reasonably well. Discrete stochastic picture. It's important to be discrete stochastic. So this is a discrete stochastic approach, similar to what we had before, a little bit more complicated. So you can view a DNA as a lattice of sites. One of those sites is a target. Your proteins are in solutions, or there's additional state. The protein could be on DNA or in solution. So there's a rate of binding to protein and binding from protein. For simplicity, you assume that DNA is a homopolymer in a typical physics way. There's a binding rate, unbinding rate, and there's a diffusion rate. Okay, and the question here is, how long does it take to find a specific target? Okay, and at time 0 you start in solution, which is full state 0, because you're diffusing so fast in 3D. The whole environment around the DNA, you call it solution, and it's a one state. And that's, as you can see, the way I posted the problem, it's perfect for first passage approach, right? You can do it through forward master equation, but it's perfect for first passage because, to remind you, the question is, I want to know if I start in solution, when for the first time I will find the target, and that will be search time. Okay, so you write these equations. We saw these equations before, right? So you see, this is Fn is a probability density to reach the target at time t. If your time equals 0, you started in one of those states. And you can see if you DNA, then you can go to state n+1 and n-1. You can also go into solution with rate KO, and that's what you can write. You can solve the things. You can also do Monte Carlo simulations here. You can solve it, and essentially something that already mentioned with you. You can get the times by going into Laplace language, and you can solve explicitly in Laplace transformation, and you can get the times. And this time is associated with search time. And it's explicitly what I needed. I needed time for starting from a solution, which I call T0. Time means search time to reach the target if I start from the solution. That corresponds to experimental conditions. And this is the essentially exact result of the calculations. It's actually easy a little bit to present it this way, and then you can explain this result even without doing mathematics. So capital L, again, is the length of DNA. Capital S is the number of sites that protein visits in one cycle. Then L over L divided by S is the number of search cycles. 1 over K on is time to be in solution between during each cycle. 1 over K off time to be on DNA. But essentially, the formula says L over S, this is how many times I have to be in solution. This is how many times I have to be on DNA. And the reason there is -1 here is because your last excursion to DNA will be successful. So this is the formula we can easily explain, and it has physical meaning. Again, this is stochastic model. You solve this model using first passage approach. That was the reason we were discussing first passage before. And there is a relevant length scale. There are several length scales in the problem. So first of all, there is this target size of order of several base pairs. There is also another length scale, which looks similar, right, to what we had before. Remember, in all previous problems, we had this time scale, square root of diffusion coefficient over dissociation rate. This is average length of the DNA scale during one search cycle. And there is another length scale, it's DNA length. So there is 3 length scales in your problem, 3 sizes. Size of target, scanning length, and length of DNA. 3 different length scales, and it leads to 3 phases, 3 dynamic regimes, 3 dynamic search regimes, depending on relative values of these length scales. And we can actually change these, and these are fixed, but we can change this scanning length by changing diffusion coefficient or changing dissociation rate. There are 3 regimes. Let us go over these 3 regimes. So what I plot here, this is the mean search time as a function of this lambda scanning length. So the first regime comes here. The first regime comes here, and I call this a random walk regime. This is the regime at which the set of parameters are such so your scanning length is larger than the length of DNA. So what's happening? Your protein comes to DNA, and it will not dissociate until it finds the target. Okay, so that I call it random walk regime. It's slow. It's slow because to move on DNA, moving on DNA is slow. So this is the first regime, which I call random walk regime. There is another regime, which I call sliding regime. This is the regime when your scanning length is larger than the target length, but smaller than the length of DNA. So you come, scan, dissociate, come back, scan, dissociate. So L over lambda, this is how many times on average you do this search cycle. And for each of this regime, I have explicit formulas. And there is one more regime, which I call jumping regime. This is the regime when your scanning length is smaller than the target size. So simplicity, I took target size as a unit, as 1. So this is the case that I told you before. You come to DNA, but you don't slide, and you dissociate. You come, dissociate, come, dissociate. So you do a lot of cycles, but this regime is different from this one. And if you do mindlessly continuum theory, which people did solving differential equations, you will get exactly the same result as I get, except over there. The continuum theory will predict these things, will predict that as lambda is going to 0, the time goes up. Exactly what I told you, diffusion coefficient going to 0 will blow up the result. So this is the prediction of continuum theory. But the point here, continuum theory cannot be applied over there. So continuum theory is, let me come back for a moment. Continuum, when you do continuum approximation of discrete phenomena, it's assumption that your lattice size is much smaller. Okay, and this assumption breaks over there. Well, fully discrete model correctly describes everything physically consistent. Not very smart use of continuum theory will do something unphysical. This is unphysical. Okay, and by the way, this is the source of this speed selectivity paradox that I mentioned. Strictly speaking, doesn't exist. It's just not very smart use of continuum approximation at the conditions when you cannot use continuum approximation.
So the continuum theory mostly fails in the calculation of the
Right. So instead of difference equation, you use continuum theory, and then you So let's go back to the equation. So you will understand. So you can even do cross passage. Yeah. This is a still difference equation. You understand, right? But now let's suppose what continuum theory does, it changes this difference into differential. Will be derivative with respect to time, derivative with respect to n, right? That's what I mean. It's okay to do it, but as long as you remember that this picture assumes that the scaling length lambda is much larger than the length of the target.
Also, is the scanning length the length?
It's a length. So protein comes to DNA to scan DNA, and that's the average distance it scans before it dissociates.
And this is by this random walk type thing on the DNA?
Yeah, it's they're doing like this, exactly, right. So it's okay to use differential equations, but do remember, so sometimes you have to remember that it's approximation. Good. So we can do the calculation. The theory is consistent. Stochastic theory consistent with all experimental observations, and essentially not only stochastic theory is successful, but it also can provide a very reasonable explanation of this phenomenon. So the idea of the fast search is that there is a strong coupling between slow 1D search and was fast 3D excursion. So there is some. So your protein comes to DNA scans, but because its non-specific interactions are not too strong, it goes away. But because this non-specific interaction exists, you want to come. So you're effectively increasing the speed of your system. Okay. Then some of you, if you notice, so this is the time in log scale. This is their scanning length, and you can ask me if I wouldn't have this 3D plus 1D search, I would have fast result would be like this. Okay, there is a little bit. There is a little bit of acceleration here, but it's not 2 orders of magnitude. Not 2 orders of magnitude. I told you about 2 orders of magnitude acceleration, but it's not here. So what is going on? There is one more component is missing. And again, so let me repeat the logic. You have phenomena, you have a model which describes things. It describes many things, but not everything, right? Then you change the model. That's the logic here, okay? That's the way you do it. And so what we're missing something which intersegment transform. That's monkey bar motion. So monkey is a good example here. So the protein is not a point, it's some object which has several hands where it can hand to. So imagine DNA is whatever it hands. So if I hand over here, I can really quickly go to far away because I'm monkey. So that's monkey bar. And that's what, and by the way, this is this like repressor protein, the one which really moves fast. You see, it's a tetramer. So and this is DNA here, it can bind DNA here, it can bind DNA here. So this intersegment transfer is additional phenomena which can be done fast. And we together with my experimental collaborator, we actually investigated this for the system. For the system where we looked at protein called EGR1 protein. So EGR1 protein is a so-called zinc finger protein, and the target this is the sequence of the target that it likes to come. So this protein has interesting two functions. It has two functions. First, it activates long-term memory. And second, this is the important protein against stroke. So if you don't want to have a stroke, train your long-term memory, okay? That's the best way to do it, because the same protein responsible for long-term memory as well as a defense against the stroke. Anyway, so that's not important for what we did. So in this experiment, the experiments were done in the following way. So they had a piece of DNA, not very long. They had this target, they put it at the beginning, and also you don't see how, but they added some fluorescent label. And so the point here, they could follow the fluorescent signal, and when protein was coming to target, fluorescent was going down in this case. So that you could really see the way experiment is done. And then my experiment collaborator had a very smart idea how to measure this monkey bar or intersegment effect. So he did the following thing. So this is your prop DNA with your target, and this found this fluorescent segment that he added there. But he also added in solution species of DNA without targets much shorter. And the idea was that if this intersegment transfer happens, so occasionally your protein can go and bind to those competitor DNA without targets. So the change in the concentration of the competitor DNA, you in some sense you can model, you can see the effect of this intersegment transfer. And we did it, and it worked reasonably well. So this is experimental proof. So effect measured rates at different conditions, and our theory, we use our theory to feed this data, and it worked quite well. So essentially, we were able to explain the two orders of magnitude acceleration. We were able to explain 2 orders of magnitude acceleration due to intersegment transfer. Okay. So this is the explanation of acceleration which comes from our simple theory, stochastic theory. So remember, we started with this result. This is the maximal possible rate of the chemical reaction can happen, which comes from the Biot-Smoluchowski theory. And remember, the wrong theory was argued that there is effective increased size. Our theory says it's not the effective increase, it's increased effective mobility. So this DNA and this interaction and this intersegment transfer, this increased effective mobility of the molecules. So the protein molecules like to come to DNA because of non-specific interaction. They can also jump between different parts of DNA due to intersegment. All of this leads to effectively larger diffusivity. And that's what, in our opinion, is the right description. And this is a description which is consistent with all observations. There is no any physical observations. The system works quite well. Okay, so good. So we're almost finished here. So this is an application of stochastic system, right? Discrete stochastic system. The importance of this application is that was an example that people could wrongly use continuum approximation and get some nonsense. Okay, yeah. There is a famous story of cold fusion. Probably young enough to remember a story about cold fusion. In 1988. It was discovered at the University of Utah that there is a cold fusion, that at relatively low temperatures you can get some essentially nuclear reaction, and it was and the answer was very simple. So it was in chemistry department in Utah. It was a wall. Beyond the wall was physics department, and they had a stove just for the current. So the stove was heating this thing, and that was this effect. Anyway, so it is important to double check, triple check what you are getting. Sometimes some of the most surprising results are wrong because you did something wrong mathematically, and that's an example here. Of course, these people were able to sell it well. It sounds interesting, but it's not always right. Anyway, so this is my conclusions here. So we developed this stochastic discrete state approach, which can explain how proteins can find targets on the DNA, how they can find it so fast. It's a combination of one-dimensional 3D motion, and there is also, it's important to take into account intersegment transfer. We found this kind of a phase diagram, dynamic phase diagram. There are 3 regimes. That's because this depends on different values of relevant length scales. And relevant length scales in this problem is 3 relevant scales: target size, scanning length, and length of DNA. Essentially, we were able to explain microscopically, more or less microscopically, the search acceleration. And this our stochastic theory resolves fit selectivity paradox. It doesn't exist. Fake news. And we did, of course, Monte Carlo simulations, and everything agree with all experimental observations. Okay. So this is the end of my lecture here. Again, I'm trying not to over. So this is another story completely different from what was before, where stochastic approach works reasonably well. And then one more lesson, that continuum is always approximation. You have to remember this. That's all. Any questions?
So in your discrete state stochastic theory, so you said that the best fit you got was using a combination of 3D, 1D plus monkey bar dynamics.
That's right.
In the 3D, 1D case, you have the hopping along the lattice, and there's also disconnecting from the lattice. Is that right?
That's correct. So in this model without intersegment transfer, you start in solution. You can bind to DNA anywhere equally probable with rate field. You can unbind from DNA somewhere, and you can diffuse with the rate field until you find the target. This is the simplest story. You always start with the simplest theory, develop as much as possible, and see if it's consistent with experiments. So it was consistent with many, but not all of them. Then you need to do something in addition.
So here, when you're in the solution, you may bind to any of the sites.
Yes.
And then you hop around the sites.
Correct.
And then you may detach.
Right.
And then, okay.
Right. So it's a almost 1D, not exactly one, but quasi 1D model. So, and that's the reason it's quasi 1D allows to do analytical solutions. But intersegment transfer, you can think about intersegment transfer that you can simultaneously jump from here to somewhere here with some rate. That's what intersegment transfer. You still can do analytical work. You still can do calculations.
So for the intersegment transfer, what I understood was that since there are many strands and the DNA is not linear, you could have hopping.
That's exactly, yeah. So it's the two units on DNA could be far away along the contour, but could be physically close.
Okay.
And that's what it is.
So you could so you still keep one strand, but you allow the possibility
Yes.
of jumping from.
Yes. In a simple thing, you allow the possibility to jump from here to here or here to here with the same rate. There's approximation because the rate's probably not the same, but it's a reasonable one. That's how we feed all this. The points are experiments. The lines are theory. The theory which adds this additional thing. So it fits quite well. There's only so we have a uniform feeding global feed with only one parameter to all data that this my experimental collaborator has. Okay.
How much is this close to if instead of taking a line like a discrete setup, if we have top of models, instead of rigid lattice, we can have some flexibility of lattice by introducing some parameters, and then they can affect the rates, hopping rates.
Yeah, so first of all, it's a single particle here. So it's a single random walk here. There is nothing around. We did extension when we have several of them. Then there are some additional features appears, but it's never the limit of the set, but density plays a role. Typical things, the concentration of this protein is quite low, so it's almost always single molecule picture. Relatively speaking, it's a single molecule.
Also, in your discrete model on that string, the DNA thing, which are the rates to go left and right are the same?
Yeah, the same. Which is also approximation, right? There's a DNA is a sequence dependent, so the rate for right left should be probably not the same, but will be very close, and on average will be the same. This is typical physics way. Sequential dependence is another effect.
And from the solution, when you attach to one of those sites, do you assume that you're equally likely to join any of the sites?
Yeah, and the reason is that experimental data tell that the diffusion in solution is fast, very, very fast. So if it's DNA, and quickly go anywhere, so that after a very short period of time, I can equally bind anywhere with equal probability. It's approximation. Mhm. Mhm. Any more questions? Probably.
Hands up.
Thank you. Is it still too much chemistry here? No. No. So it's my main reason I wanted to present this to show that continuum sometimes could be wrong. But we're talking about differential equations, right? Which is right.